Welcome again to the YouTube live classes for botany for class 12. Today we will start a new topic and most interesting topic, that is linkage and crossing over. Now, as we have discussed the Mendelian inheritance and the deviation from Mendelism, so this is one of the deviation from Mendelism that we are going to discuss. So, I am Dr. S.S. SARDAR from RD Women's HSS is going to deliver the topic of linkage and crossing over. So, let us have a look on linkage. So, what is the definition? The definition of linkage is the tendency of two or more number of genes to be linked together and to be inherited as a single unit. So, it is a tendency of two or more linked genes to inherit together in the same combination for at least more than 2 generations is called linkage. Now, the discovery is credited to William Bateson and Reginald Punnett while working on sweet pea or Lathyrus odoratus, but could not explain the phenomenon properly. So, who explained the phenomenon properly? It was Morgan, T.H. Morgan in 1911 while working on Drosophila melanogaster. He confirmed this theory that some traits are linked together and they are inherited together as a single unit. Okay? Whereas the Mendelism it tells now the different trait or a single trait is independently inherited from another trait. Whereas this linkage theory it establishes that the traits can also be linked together and they can be inherited as a single unit or something like that. So, let us have a look on this discovery. Now, George Mendel's law of independent assortment states that every trait is inherited independently of every other trait. So, what the law of independent assortment says, that every trait is inherited independently from another trait. So, here linkage or here the first experiment of Bateson and Punnett on sweet pea, which could not explain the theory exactly, but they said that this is somewhat a kind of coupling and repulsion theory that we are going to discuss in the next slide. Now, because of this coupling and repulsion by Bateson and Punnett while working on Lathyrus odoratus or sweet pea, now they proved that some of the traits they are linked together. Okay? So, that is why the concept of linkage, it nullifies the universality, of the law of independent assortment. So, the law of independent assortment is not universal because of the fact of linkage and crossing over. So, coming to the next point, but shortly after Mendel's work was rediscovered, exceptions to this rule were found. In 1905, the British genetist William Bateson and Reginald Punnett, while working on the same line of experiment as Mendel, discovered a concept of linkage, although they could not explain it. Now, they were interested in trait inheritance in the sweet pea and were studying two genes. One is the gene for flower color, the other one is the gene affecting the shape of pollen. So, the flower color is of two types, one is purple, the other one is red. So, purple is dominant, the red is recessive, and the pollen grain long is dominant and the round is recessive. So, let us have a look on the experiment. Now, what did they do? Now, they first of all they crossed a blue flower long pollen with a red flower round pollen. So, they crossed a blue flower long pollen with a red flower round pollen. So, these are dominant characters, okay, and these are recessive characters. So, these are recessive characters and these are dominant characters. Now, when they cross the dominant for both of the traits with the recessive for both of the traits, now in the F1 generation they got blue long flower. That means blue flower and long pollen. So, blue flower and long pollen they got. Now, this is the dihybrid test cross. Now, when they have gone for a test cross by using this F1 progeny or F1 generation progeny with the recessive parent. Now, what they got? They got 4 different combination as is expected. Now, 2 of them will be parental like blue flower long pollen and red flower round pollen, whereas the other 2 obviously will be the recombinant type or it would be a new combination type. So, what it would be? It will be blue flower to round pollen and red flower to long pollen. So, we have got it here, the blue flower long pollen and red flower round pollen as the parental character, whereas the blue flower to round pollen as I have crossed it here. So, blue flower to round pollen and red flower to long pollen. So, these 2 are what? A new combination. But what did they found? The test cross should be 1:1:1:1 ratio. It should come. The outcome should be in this ratio or the expected ratio should be this if it would be properly following Mendelism or the law of independent assortment. But here the observed ratio was shown to be 7:1:1:7. Okay, that means the parental combinations they are coming at maximum numbers or same but maximum. So, blue long and red round they are coming in ratio of 7 or in percentage of 44. Okay, whereas the blue round and red long which are sorry recombinant type they are coming at the percentage of 6%. Okay, so this says now they couldn't do it, then again they designed the experiment in a different manner. So, what did they do? Now, they in the second experiment they chose, so let us go to the second experiment. So, in the second experiment now you see what they did, they chose blue flower is dominant but round pollen. Now, round pollen is recessive. So, what did they do? Now, they do first of all they cross a dominant trait or they combine a dominant trait and a recessive trait. So, the blue flower is dominant, round pollen is recessive and red flower is what? Recessive and long pollen is dominant. So, what did they do? Now, they took the recombinant type as the parental combination. Now, what is the parental combination here? The parental combination is blue flower to round pollen and red flower to long pollen. Now, you can see the genotype here. Now, the genotypes are being capital B, small l, and small b, capital L, capital L. So, obviously, this will form a gamete like capital B, small l and here a small b, capital L. So, the F1 generation, all the plants that we are getting, they are in blue and long. So, the similar result we found as we have found it before. So, like this. So, as we have found that all the F1 generations are blue long in the parental combination where the 2 dominant traits are taken together and 2 recessive traits are taken together. Now, here also we have got the same result. So, what should be the expected ratio for the F2 test cross generation? So, it will be somewhat like the previous one, okay, as it was shown in this previous slide, but here they got something a different kind of the same ratio, but in a different combination. Now, what is the combination here? Now, what is the parental combination here? Now, this is blue round. So, blue round is the parental combination and also red long. So, the red long, it has been, so here the red long parental combination, this is not round. So, red long because you see here blue long, red round. So, here this is red long. So, the red long is coming maximum, or in other words, let's have a, just a minute. Okay, now you see, now the F1 generation is blue long. Now what is the ratio? Now here the ratio changes as 1:7:7:1 instead of the expected ratio, that is 1:1:1:1. Okay, so here you see the parental combination is blue round, red long. So this two will come in maximum numbers. Okay, so let's have a look. So the blue round and the red long, they are coming at maximum number as 44% or in the ratio of 7:7. Whereas the dominant traits, or this will become the recombinant type or not. So this will become the recombinant ones. Now you see this two. So the blue long and red round will now be the recombinant types. Now they will come as 1, 1 or the percentage frequency as 6 and 6. Okay, so here they explained in a different manner that the blue round and red long are parental forms and so greater frequency of 88% and blue long and red round are recombinant forms. Okay, and so lesser frequency of 12%. So let's have a look together. Now this is the coupling phase, this one is the coupling phase and this one is the repulsion phase. So togetherly you see here the parental combination is blue long and here it is red round. Here the reverse, okay, blue round is the parental combination and red long is the parental combination. So here we can see or we can observe one thing in common, that the parental combinations, they are coming in maximum ratio in the F2 test cross generation, whereas the recombinant types, they are coming at minimum numbers in the F2 test cross generation. Okay, so how could they explain it? They explain it in a manner that here the dominant alleles, they remain together. That means in the first experiment or in the coupling phase. Now in the coupling phase, the dominant alleles, they remain together. The recessive alleles, they remain together. Okay, and as such in the F2 test cross generation, they are coming at maximum numbers, blue long and red round. And the recombinant types, if we will go for a recombinant types, they are coming at minimum. So here the dominant traits or the dominant alleles are coupled together and the recessive alleles are coupled together. But when we are taking it another way, that the dominant and recessive, one dominant, one recessive alleles for two traits, now they are repelling each other. Now here the dominant and the dominant alleles, they are repelling each other and the recessive alleles, they are repelling each other. So in the coupling phase, they said that the dominant and recessive alleles, they are coupled together. In the repulsion phase, they told that the dominant and recessive alleles, they are repelling each other. Okay, so they couldn't explain the exact phenomenon, what is happening behind. Well, so it was actually established, or it was actually put forth, or it was established by Morgan. Okay, so the Morgan theory of linkage is best known, and he explained the theory of linkage very well while working on the fruit fly, or in other words Drosophila melanogaster. So the phenomenon is explained by T.H. Morgan in 1911 while working on Drosophila melanogaster with, now he found that it has a grey body and long wing and a black body and vestigial wing, two different types, and this combination is, they are coming always. Now he found a complete linkage. So from this he told, or he proposed that the traits can also be linked together and can be inherited together as a unit. So he put forth the chromosome theory of linkage. What it says? Let's have a look. Now the linkage theory says according to Morgan and Castell and other scientists, that they concluded that chromosome bears many genes. Now, genes are arranged linearly on chromosomes. Okay? Now, chromosome bears many genes. Genes are arranged in a linear fashion. Then, the genes which show linkage are situated in the same chromosomes and are bounded by the chromosomal material. So, I will show you by drawing the diagram. Now, the strength of linkage, that means how two genes will be linked together and will be inherited as a single unit, it is decided by it is decided by the distance present between these two genes. Okay? If the distance is closer, the two genes will act as a single unit and will be inherited from generation to generation as a unit. If the distance increases, now the strength of linkage, it decreases. Okay? So, the strength of linkage depends upon the distance between the linked genes in the chromosome. This is another concept, and the linked genes remained in their original combination during the course of inheritance. Okay? As he found it in this Drosophila melanogaster, a complete linkage that we are going to discuss in the upcoming slides. Now, in this regard, now what he found is a complete linkage. Now, linkage can be of two types. One is a complete linkage, the another one is incomplete linkage. So, the complete linkage is that the two traits or the genes, they are always linked together. That means the distance between these two are so less that they cannot, the process or the phenomenon of crossing over cannot happen there, and they will always be inherited as a single unit, although these are two genes and they are present on two different loci, but the distance between them will treat them as a single unit and they will be inherited from one generation to another generation. So, this is the concept of complete linkage, whereas in incomplete linkage you will find that apart or along with the parental types, now you will get recombinant types, but in complete linkage now you are not going to get any sort of recombinant types. We will discuss it further with the help of examples. Okay. So, before going to that, let me tell you that how the linkage or the linked genes are present on the same chromosome. For example, let us suppose this is a chromosome. Okay, this is the centromeric region. The genes are arranged linearly like this. Okay, let us suppose there is no crossing over between the homologous chromosome. Now, we will have a similar kind of chromosomes also having the same loci and all. Okay. So, if there will be no crossing over at any point between the loci, now they will be inherited as such. So, as many number of genes that are present, okay, as many number of genes that are present on this chromosome, they will be inherited as a single unit. But this is a very rare phenomenon because crossing over happens between two homologous or between the homologous chromosome. So, these are homologous chromosome, and because of this, so it happens between homologous chromosome, and because of, let us suppose anywhere the crossing over takes place. So, it will it will alter the combination and we are going to get new combination or recombinant types. Okay. So, that is why the linkage or that is why the genes which show linkage are always situated on the same chromosome. We cannot say that if this is a homologous chromosome, another chromosome is like this. Sorry. This, okay, having few number of genes. Now, a gene from this chromosome and a gene from this chromosome cannot be linked together. They can never be linked together because they are present on different chromosomes. Okay. So, So, let us have a look on the complete linkage. So, what it says, complete linkage or linkage of genes on a chromosome which is not altered and is inherited as such from generation to generation without any crossover is called as complete linkage. Now, in this type of linkage, genes are closely associated and tend to remain together, whereas it is defined as the state in which two loci are close together and the alleles of these loci are virtually present and virtually never separated by crossing over. Let us have a look on the example. So, this is the example while T.H. Morgan while walking on, now he had chosen two parents, one is grey body color and long wings or simply grey body long wings, and the another one is black body and vestigial wings. Now he found, now when he crossed it, now all the F1 generation he found that all the progeny they are all grey and long as like this. So this is the dominant one and this is the recessive one. Okay. So in the F1 generation he is going to get all the progenies as grey and long. Now when he has gone for the test cross in the F2 generation, when he has test crossed it, now he got two combination again in the form of grey long body and black vestigial body. But for your kind information, there is no recombinant types. That means what? Now the grey body long wings, it is a parental combination. Okay, this is parental and this is parental. If you would have got some recombinant types, now what it would have been? This would be like grey vestigial and what? Black long, black to long. But these two recombinant types he never found throughout his experiment. So what he got is 50% grey long and 50% black and vestigial. These two combinations are parental type. So as per the parent, so these two are parental, but he never found any kind of new combination or recombinant types. So from this he concluded that the body color and the wing shape, okay, this or the wing type, these two are linked together. Well, so because of the strong linkage, he always found this parental combination, no recombinant types. So what he explains? He explains that ordinary male wildfly with grey body and long wings or normal wings was crossed with female having black body and vestigial wings. So in the F1 hybrids, all are grey bodied and so all are grey bodied and normal wing. Now when he crossed the F1 generation progeny with, or when he has gone for a test cross for this F1 progeny, in the F2 test cross generation he found the same two parental combination as grey body and normal wings. So what he found in the F2 generation? So these two types were again in the F2 generation, grey body normal wing and vestigial black body and vestigial wing. Now you see the F2 generation population are similar to the parental combination. So this shows the complete linkage between these two traits, okay, and which are inherited as a single unit from generation to generation. Whereas incomplete linkage, so linkage are of two types, one is complete linkage, the other one is incomplete linkage. So the incomplete linkage produces always, what is the characteristic feature of this incomplete linkage? It is always, it produces new combination along with the parental types. Now what can be the example? The example is by the experiment on sweet pea or Lathyrus odoratus by Bateson and Punnett. Okay. So, it says that incomplete linkage produces new combinations of the genes in the progeny due to the formation of, and why it happens? Because of the formation of chiasma or crossing over in between the linked genes present on the homologous chromosome, that we have discussed already. Now, the genes which are present or the linked genes which are present, they are always in the same chromosome, and the new combination, it comes from the crossing over between the homologous chromosomes. It is not that any two chromosome will come and it will cross over. This is not like that. As we have read this cell divisional process, we know the process of crossing over, and crossing over only takes place between two homologous chromosomes. Okay. So, when in sweet peas, now what is the example once again? Now, in sweet peas, the blue flower and long pollen, when they are crossed with the red flower and round pollen in F1, now the F1 is expected blue flower and long pollen heterozygous condition is got, and when they have gone to the F2 test cross generation, now they got these two parental combination, along with that the blue flower to round pollen and red flower to long pollen. So, this is an example of incomplete linkage. For your kind information, most of the linkages are what? Incomplete linkage. The complete linkage is very rare, okay, as we have seen it in case of this grey body normal wings and black body vestigial wings of Drosophila melanogaster. Now, what is the significance of linkage? Now, the significance is linkage does not permit the breeders to bring the desirable characters in one variety. If the linkage is very strong, now it cannot go further, or it cannot go for new recombination. So, it is creating problem for the plant breeders who is desiring to get new combinations for better improvement or for the improvement of the crop. Now, next, link characters are maintained for generation because linkage prevents the incidence of recombination. Now, next point is linkage groups. Now, what are linkage groups? For example, let us have a look on this. Now, Drosophila melanogaster, now it has the diploid number of chromosome as 8, or we can say that it has 4 homologous chromosome or 4 pairs of chromosome. 4 pairs of homologous chromosome. Okay, 4 pairs of homologous chromosome. So, here now you see, now all the 4 chromosomes, now if we will consider a haploid number, now within the cell, these haploid chromosomes, I have drawn only the haploid. Okay, these 4 chromosomes which are haploid in nature, now they constitute a linkage group. Now, how many linkage groups are possible in Drosophila melanogaster? It is the haploid number of chromosome. Now, what is the diploid number? The diploid number is 8. So, the haploid number is 4. Now, 4 numbers of linkage groups are possible. Okay. Now, for example, in human beings, how many chromosomes are there? Now, there are 46 chromosomes, or the diploid chromosome number is 46. Now, exactly we have 23 pairs of chromosome. Now, how many linkage groups are possible in this case? Because it is the haploid number is 23. So, 23 linkage groups is possible in case of human beings. So, as in pea plants, now 2n is equal to 14. So, 7 numbers of linkage groups are possible in case of pea plants. Why it is so? Because you see, now here these are the 4 chromosomes, 1, 2, 3, 4. Now, here this is a linkage map, that how the different morphological traits, they are being present in combination to another. That means here, as per the recombination frequency and the linkage between the genes, okay, they have been designed, or they have constructed a linkage map here. So, it says that this kind of traits are present on the first chromosome or on the X chromosome, okay, then these traits are present, and this numbers this says about the recombination frequency. That means this is not a physical map how the genes are present on the chromosome, rather this is this is based upon the linkage between the genes and its recombination frequency by means of a 3-point cross. So, here you see now all these genes present together, now they constitute a single linkage group, number 1. Now, here it constitutes another linkage group, number 2. Now, here all the genes which are present here, now they constitute a third linkage group, and here it comprises of a fourth linkage group. So, in this regard it has 4 numbers of linkage groups because of the presence of 4 numbers of homologous pair, or the haploid number is 4. Okay. So, the linkage group is that all of the genes on a single chromosome constitute a linkage group, as we discussed here. They are inherited as a group, that is during cell division they act and moves as a unit rather than independently. And finally, the existence of linkage group is the reason some traits do not comply with Mendel's law of independent assortment. So, the law of independent assortment is because of the presence of the phenomenon of linkage. Okay. So, what is a linkage map? Now, as I have shown you here, now this is the linkage map. So, this is a linkage map. It says the position of the genes in terms of recombination frequency. This is actually not a physical map. Okay. So, what it says that a linkage map is a table for a species or experimental population that shows the position of its known genes. Okay. The known genes or genetic markers relative to each other in terms of, that I told you, in terms of recombination frequency rather than a specific physical distance along each chromosome. So, let us have a look that what are the factors that affects a linkage map. Now, the factors, obviously the distance between genes, age, temperature, X-ray, etc. are the factors that actually influences or it affects the linkage. Now, how? Now, if the distance between 2 genes it increases, it decreases the strength of linkage. Now, with the increasing age, the strength of linkage decreases. With the increase in temperature, the strength of linkage decreases, and with the irradiation of X-rays, again the linkage strength decreases. Okay. So, this is all about linkage. Next, we will go to crossing over. Now, before going to crossing over, let me tell you one thing. Linkage and crossing over, they are the 2 sides of the same coin. If the distance between the linked genes are less, now they are inherited as single unit. If the distance increases between the linked genes present on a single chromosome, now they show crossing over, and because of crossing over, we are getting recombinant types. So, linkage and crossing over, they are very much complementary, or they are very much related to each other. Okay. But whereas the phenomenon is different, whereas linkage in linkage the genes are linked together always and are inherited as a single unit, whereas in crossing over these linked genes they found a new combination. So, what it says? It is the exchange of chromosomal. So, what is crossing over? It is the exchange of chromosomal segment between homologous chromosome and because of the formation of chiasmata. Okay. So, it is the exchange of chromosomal segments between the non-sister chromatid, okay, of a pair of homologous chromosome. Now, a pair of homologous chromosome or a single homologous chromosome, if we consider, now at the time of cell division, now it will be in duplicated phase and it will have 2 sister chromatids. Okay. So, crossing over, it always takes place between non-sister chromatids. Well, and the term was introduced again by T.H. Morgan and Cattell in 1912. So, when they found linkage in 1911 and crossing over in 1912. The crossing over occurs at which stage? It occurs at Pachitin stage of the prophase 1 of meiosis. Okay. So, it occurs at Pachitin substage of prophase 1 of which one? Of meiosis. Because as you know, mitosis does not have any two phases like prophase 1 and prophase 2 like this. So, it is in meiosis where this Pachitin, at the stage of Pachitin it happens, this crossing over happens. And how it happens? It happens because of formation of a special kind of structure known as chiasmata. So, chiasmata are the points at which crossing over takes place. The chiasmata were first discovered by Janssens in 1909. Now, the number of chiasmata is variable. That means, let us suppose if I will draw that if this is a chromosome and these are the homologous pair, okay. If the crossing over happens between these two, well, now the chiasmata that is formed, now here one chiasmata may be formed, again another chiasmata may be formed between another two loci, okay. So, chiasmatas are variable, that means for a single number of homologous pair or homologous chromosome pair, now it may be more than one or it may be 1, 2, 3 like this, but generally the minimum rule is that a chiasmata will be formed for a single homologous pair of chromosome. So, here the number of chiasmata is variable, minimum one chiasmata per bivalent is the rule, but as many as 12 per bivalent is seen as Vicia faba. Okay. This is one of the member of Fabaceae family. So, the non-sister chromatid in which exchange of segment takes place is called as crossovers. Now, always the exchange takes place between two non-sister chromatids or recombinants, they are called as crossover or recombinants, and the other two which are not involved are called as non-crossovers or parental type. Now, what it says, now let us have a look. Now, let us suppose okay, this is one homologous chromosome and this is another. Okay. Now, this is let us suppose 1, this is 1 prime and these two are homologous chromosome. Okay. Now, what happens? It always happens between the non-sister chromatids. If I will number it as A and B and this is A prime, B prime. So, it happens between these two or like this two like this, okay, between A, B prime or A prime, B, A prime or B, B prime, okay, not between A and B and A prime and B prime, okay. So, it always happens between non-sister chromatids. Now, which one will be considered as crossovers or recombinant? Now, when there is a crossing over between these two, now these two non-sister chromatids after the crossing over products, now they will be considered as crossovers or recombinant types, whereas this one and this one, okay, A and B prime, they will be considered as parental combination, okay. So, mechanism of crossing over, now we will go for a diagrammatic by doing the diagrams. Now, the mechanism of crossing over is that, now one is synapsis, then the second one is 4-stranded stays, crossing over and then terminalization. Now, let us have a look on the cell divisional process. Now, in the cell divisional process or in meiosis, now the first phase is the karyokinesis or the first phase is prophase 1, then your metaphase 1, then anaphase 1 and telophase 1. Now, in prophase 1 we have 5 substages. Now, what are this? These are leptotin, zygotin, pachitin, diplotin and diakinesis. Okay. So, in the first leptotin, now you will see that there will be appearance of chromosomes which will come in pairs, and the second phase leptotin then zygotin, In the phase of zygote, now the synapses occurs. In the zygote substage, now where it happens? It happens in prophase 1. Okay. So the synapses happen. So what it says, now let us suppose this is a chromosome. Okay. So always it comes as 2 sister chromatids for this chromosome. If I will draw another chromosome for this homologous pair, so these 2 are homologous pair. Okay. So now this is 1 prime. Now the crossing over happens between, or first of all in the stage of zygote, now synapses occurs. What it means, now the 2 homologous chromosome of a pair, now they come near each other, okay, in the 4 stranded strands. Now you see this has 2 strands, 2 sister chromatids, the other one has 2 strands also. So they are in 4 stranded stages, okay. Then crossing over takes place. Now crossing over means if this is one loci and this is another loci which are close to each other, which are linked or as a chromosome, now several loci will be there. So when any 2 will be formed, a chiasmata. So here a chiasmata will be formed, that is a complex structure, and there will be exchange of chromatids between them. Okay, there will be exchange of chromatids between them, and finally we will get a crossover product. How I will tell you in the next diagram or by doing the diagram. And finally this will slowly, this will come to this terminus for terminalization, and the chiasmata will move from this to this end, and finally it will be removed out. So it will be removed out and we are going to get the crossover products. So the types of crossing over is like single crossing over, double crossing over, multiple crossing over and somatic crossing over. Let me show you one single crossing over type, that let us suppose this is a chromosome pair. So always, as you know, the remaining 2 strands. So this is one prime chromosome. Now here the genes are like A, B, C, A, B, C. Now here it is capital A, capital B, capital C, capital A, capital B, C, okay, as it is in duplicated form. So in a single crossover there will be a chiasmata that will be formed for at a single point. Okay, so sorry. So no, so this is not, actually there will be a chiasmata that will be formed between these 2. Okay, so a chiasmata will be formed between these 2. Okay, now if these 2 are like so, crossing over, it always happens between 2 non-sister chromatids. Now this is a single crossover. So what are the products we are going to get is like the parental combination as ABC, then the recombinant combination as here A, capital B and small c. Okay, this is the recombinant type. Then again here we are going to get small a, small b and capital C. Well, and finally small a, small b and small c. So these are the 4 kind of combinations, and we are going to get the single crossing over, double crossing over, multiple crossing over and somatic crossing over. Now The theories of crossing overs are of two types. Now one explains the two plane theory, that is the classical concept by L.W. Sharp, and another one is one plane theory, that is the chiasma type of theory, that is proposed by F.A. Janssen and fully developed by Belling and Darlington. Okay. Now these are the theories, and what are the factors that influences crossing over are distance, age, X-ray, sex, temperature, heterochromatin, chemicals and interference. Well, and what is the significance? The significance, it produces a new combination of genes. Crossing over plays an essential role in the process of evolution. The crossing over frequency helps in the construction of genetic map, and it provides the evidence for a linear arrangement of linked genes in a chromosome, and finally it provides an inexhaustible store of gene variability in sexual reproducing organisms. So these are the significances of crossing over, and finally we have covered linkage and crossing over. Thank you very much.